we're going to summarize all the events that took place in the cell cycle, and we will correspond it with the fluctuations in the amount of different cyclins. Whenever a cell is exposed to some growth factors, so it will upregulate the amount of cyclin D that is present in the cell, so that it will increase the transcription and ultimate translation of cyclin D1, cyclin D2 and cyclin D3. Upregulation of D1, D2, D3 depends upon the different agonists or the different receptors and the secondary messenger molecules they are associated with respectively. What we see right here is, if this is where the cell cycle starts, just about before beginning the G1 phase, so we will have a gradual increase in the amount of nuclear D1 that is present. And if we here have the R point, if this is the R point, as we saw previously, this is the period where we need those growth factors, where we need some mitogenic signals so that the amount of cyclin D1 is upregulated, which will further initiate other cascades. So now after the upregulation of cyclin D1 has happened, so cyclin D along with its CDK, that is CDK 4 and 6, they will act like a sponge and they will take up all the amounts of inhibitory CDKIs that are present, like P27. So they will soak up P27 and they will sequester it. Now, P27 was essentially inhibiting cyclin E and preventing its association with CDK2. So once P27 is sequestered, we will again have a rise in the amount of cyclin E and correspondingly CDK2 complexes that are being formed. And that will ultimately lead to the first G1S to transition. So we accomplished G1 to S transition through the upregulation of cyclin E and CDK2. This cyclin E can further initiate a feed-forward cycle, that is, the upregulation of cyclin E and CDK2 will activate transcription factors that can lead to the upregulation and transcription of more cyclin E and CDK2. So once cyclin E and CDK2 start decreasing, we will have a upregulation in the amount of cyclin A that will first associate, cyclin A will first associate with CDK2 initially, and then it will associate with CDC2, and we can call it CDK1. So throughout the S phase, the SCDK, that is the cyclin A and CDK2, will maintain and will check for any DNA damage that has occurred during replication. As we proceed into G2, we can see that there is an increase in the amount of cyclin B. So cyclin B increases, it forms a complex with CDK1, and that will be the G2M transition cyclin CDK complex. Now what? After we have done all this and we know that cyclins and activated CDKs are essentially serine threonine kinases. So what will they do? So now we have the role of something known as PRB, that is protein RB, that is the retinoblastoma protein. Retinoblastoma protein has also been lovingly being called the governor of proliferation, right? So it basically checks the amount of proliferation that a cell will undergo. So how does it do that? So now, before continuing and studying what the PRB actually do, we need to see that the PRB actually exists in three forms. It exists in an unphosphorylated form, it exists in a hypophosphorylated form, and it exists in a hyperphosphorylated form. Now, the usual job of PRB is to act like a pocket protein. Just like you put a coin inside your pocket, PRB puts E2F in its pocket and it sequesters it. E2F is sequestered by PRB, and E2F is a potent transcription factor. This sequestration is only present whenever the PRB is hypophosphorylated. So we will say the unphosphorylated, if even the hypophosphorylated state of PRB is active. So the unphosphorylated state of PRB is usually found in the quiescent stage when the cell is in its dormant stage. Now, whenever we are exposed to some mitogenic signals, and we saw the window period in which we can do that before the restriction point, we will see upregulation in the amount of cyclin D and CDK4/6. So cyclin and CDK complex that essentially phosphorylates PRB into its hypophosphorylated state. Hypophosphorylated state is present in early G1. Now, as we saw during the G1S transition, we get an upregulation in the amount of cyclin E and CDK2 that form the G1S transition CDK cyclin complex. Now, what they're going to do is they're going to again phosphorylate PRB and convert it into its hyperphosphorylated form. But there is a catch. That catch is that cyclin E and CDK2 cannot do this job without the help of cyclin D and CDK4 and 6. You may ask why. Now, the thing is, cyclin E and CDK2 does not phosphorylate the unphosphorylated state of PRB. It only phosphorylates the hypophosphorylated state of the RB, right? So we need cyclin D and CDK4 and 6 to convert the unphosphorylated state of RB initially into the hypophosphorylated state of RB, and then eventually cyclin E and CDK2 will do the rest of the job, not without the help of cyclin D and CDK4 and 6. After the cell has underwent through whole of mitosis, what we shall get it after the end of the mitotic phase, we will have activation of the protein phosphatase 1 protein that will cleave phosphate groups present on the RB and convert it back into its unphosphorylated state. We have seen how mitogens can upregulate the amount of cyclin D and CDK4 and 6 and they become active. Now this PRB, when it gets converted into a hypophosphorylated state, now it gets inactive, and the further the job is done from converting it into hypophosphorylated into the hyperphosphorylated state by ECDK2. Now this RB is completely inactive, and as we saw, the affinity of ECDK2 is just towards the hypophosphorylated state, not towards the unphosphorylated state. So we still need the mitogenic signal and the corresponding increase in cyclin D and CDK4 and 6 to do that initial job. So as we saw, again, just a repetition, we saw that the program influenced by extracellular signals is just up to the point of the R point. Now, as we studied, R point signifies two things. It signifies that the cell has become independent. It has become independent and will not require growth signals. The second thing corresponds, it basically corresponds to a major biochemical alteration that has taken place. As you see, after the R point, that is, the RB gets converted into its inactive hyperphosphorylated state. So this is the biochemical alteration that we're talking about, that RB initially active will get converted into RB that is inactive. Let's tie this all together that we have studied till now. So as we saw, we have basically some mitogenic signals. They could be in the form of receptor tyrosine kinases, they could be in the form of Janus kinases or JAK kinases, or they could be in the form of even interleukin receptors, which can promote cellular proliferation and differentiation. At first, we have the receptor tyrosine kinase pathway. So receptor tyrosine kinase pathway, we have the activation of growth receptor binding protein, just an example, that will further activate SOS, that is son of 7-less, that will further activate RAS, RAF, MEK, and this pathway, we call it the RAS, RAF, MEK, ERK pathway, in which we have serine, threonine, and serine, threonine, tyrosine kinases, that is RAF. So ultimately, what we shall get at this pathway, we shall cover in another video. So what we get out of this pathway is we have activation of ERK. This ERK translocates into the nucleus and upregulates to transcription of c-MEK, which in itself is a transcription factor that will enhance the levels of cyclin D2. So as we saw previously that we discussed about the redundancy of cyclin D and other cyclins as well. So this is the reason why that redundancy come into place, that is to differentiate between different signals, different mitogenic signals originating from the extracellular space. Now this ERK can further activate C-phos and C-Jun. They form a dimer known as a adapter protein 1. Now this adapter protein 1 can further lead to enhancement of transcription of cyclin D1. Right here we have beta-catenin, which is usually associated with cadherin, that is E-cadherin. It dissociates from the E-cadherin and is usually without any inhibitors of the destruction complex. It is ubiquitinated and ultimately degraded in the proteasome. But whenever an inhibitor of the destruction complex is present, that ubiquitination and ultimate degradation is prevented, and the beta-catenin translocates into the nucleus of where it promotes the transcription of the transcription factor, that is T cell factor, or we also call it lymphoid enhancing factor. Now this TCF and LEF can further promote the transcription of cyclin D1. And at last we have two more pathways which can do this, that is the JAK pathway. Now as we know, Janus kinase pathways, in which we have intrinsic tyrosine kinase domains, they will phosphorylate these tyrosine domains and will further lead to recruitment of STAT. STAT stands for signal transduction and activator of transcription. Now the STAT gets phosphorylated by the tyrosine kinase residues and it ultimately translocates into the nucleus where it can further activate cyclin D3. And another example which I like to discuss would be of interleukin receptors, which are usually associated with TRAF6, that is TGF beta regulatory associated factor 6, which ultimately leads to the promotion of NF-kappa B, that is nuclear factor kappa light chain enhancer of B cells. Now, this NF-kappa B goes inside the nucleus and will lead to the upregulation of cyclin D3. Now, summing it up, we have multiple mitogenic stimulus. For example, we have cyclin D1 that was getting upregulated by C-FOS and C-Jun, and the TCF-LEF that came from the beta-catenin. We have cyclin D2 that was getting upregulated by C-MEC, that through the RAS-RAF-MEC-ERK pathway. Then we have cyclin D3 that was getting upregulated by the STAT pathway, that is the Janus kinase STAT pathway, and some interleukin receptors which transfer their signal through the NF-kappa B pathway. Once these cyclin D, as we discussed, cyclin D get promoted, they will form a cyclin D CDK4 and 6 complex. Now, this complex essentially functions like a sponge. Like a sponge, it will soak up all of the present, in this case we are showing KIP1. So it will soak up all of the present CDK inhibitors. The sequestration of the P27 in this case will lead to a disinhibition of cyclin E and CDK2 activity. Cyclin E now can associate with cyclin dependent kinase 2. When the cyclin dependent kinase 2 goes inside the nucleus, it will lead to an increase in expression of cyclin E1. Now, as you can see, this is a feed-forward cycle. Now, the cyclin E1 production will ultimately lead to more formation of cyclin E and CDK2 complexes. After the expression of cyclin E CDK2, which we know functions as a G1S, this CDK2 and cyclin E complex will lead to phosphorylation of PRB. Now, as we studied previously, we have PRB which exists in 3 phases. The PRB was first getting phosphorylated by CDK4 and 6 and cyclin D complex. Now, it is getting this hypophosphorylated PRB gets more phosphorylated by the action of cyclin E and CDK2 complex. So this PRB gets hyperphosphorylated at the end of restriction point. Now, as we have crossed this restriction point, what will lead to is that the pocket protein, that is PRB, will not be able to bind to the E2F through its binding domain, so that the E2F transcription factor gets free. This E2F transcription factor has multiple roles. It leads to upregulation of DNA precursors. It synthesizes DNA precursors and lets the cell prepare for the upcoming DNA synthesis phase, that is the S phase. It leads to upregulation of cyclin E and CDK2 as well. That is, it will stimulate more production of cyclin E. Again, it is a feed-forward cycle. And E2F will also lead to upregulation of histone acetylase. And as we acetylate the histone residues on the chromatin, that will lead to unfolding of the chromatin, and we will escape transcriptional inactivation that was earlier facilitated by methylated or even unacetylated histone residues. So that is one thing. And then E2F can also lead to transcription of more than about 500 genes. So after we have synthesized the DNA, that we have crossed the S phase, do we need that same amount of E2F activity? We don't. We need to inhibit the amount of E2F activity, and for doing that we have the role of cyclin A and CDK2. And amongst the various genes that get upregulated, we have cyclin A as well. Now, cyclin A will form a complex with CDK2, that is the SCDK, and after the synthesis phase has occurred, we need to downregulate the amount of E2F present. So this cyclin A CDK2 complex will lead to phosphorylation of E2F, and after the E2F gets phosphorylated, we will have loss of transcription activating abilities of E2F. That is it. As we have seen, we first compared the cell cycle to a simple clock. We saw how the clock is getting regulated, how the clock cannot unwind, and what are the basic factors that lead to ticking of the clock. We discussed this all previously, and that is pretty much it about the cell cycle.